This is the lecture session of drugs and pharmaceuticals. In previous session, we discussed analgesics, which are painkillers and are very well known group of drugs. That includes aspirin, acetaminophen and ibuprofen. In addition to their analgesic properties, ibuprofen and aspirin are members of the non-steroidal anti-inflammatory group of drugs and are thus used to reduce swelling and inflammation. In this lecture session, we will be discussing antimicrobial drugs under this category, antifungal agents and antibacterial drugs. Let us start with the microbes. What are microbes? Like bacteria. Bacteria is a prokaryotic cell which have no nuclear membrane, no chlorophyll and cell wall in addition to their cell membrane. Many bacteria can be assigned to one of two major groups based on the Gram staining reaction. The difference in cell wall structure and composition divide them. First one is Gram positive bacteria and second one is Gram negative bacteria. We will concentrate on the cell wall both as target as well as a resistance factor. Fungi and bacteria can be saprophytes who live on dead, but many others are parasites. Some are human parasites. We mostly coexist happily with these microbes, but they can proliferate on or in the outermost layer of skin, which cause irritation of growths and in extreme cases serious infections. So, there are some effective chemicals which works against microbes like phenols. Phenols work by destroying the selective permeability of cell membranes. Quaternary ammonium compounds are also those chemicals which are effective against microbes. They work by concentrating at the cell membrane and dissolving lipids in the cell walls and membranes. Aldehydes. Aldehydes work by affecting protein structures, and ethylene oxide, it work by destroying DNA and proteins. It is a gas used for chemical sterilization. Let us discuss some antimicrobial agents like antiseptics. Antiseptics are formulations that can be safely applied to the skin, like iodine, a tincture which is ethanolic solution, and potassium iodide. Like disinfectants. Disinfectant again a formulation where the agent is too toxic or corrosive for topical uses, that is domestos, means kills of all known germs dead. We use hydrogen peroxide for this. Let us discuss the history of antibiotics. Paul Ehrlich, a German medical doctor and a researcher who is known as the father of immunology and chemotherapy. Ehrlich hypothesized that certain dyes could selectively stain harmful bacteria cell without harming host cells. He referred to such compounds as magic bullets and coined the term chemotherapy as a general descriptor for chemical remedies targeted to selectively kill infectious cells. There are different types of antimicrobial drugs. Let us classify them. The different antimicrobial drugs are antibacterial drugs which kill bacteria, antiviral drugs for killing virus, antiparasitic drugs which kill parasites, and antifungal drugs for killing fungus. So, antibiotics are those molecules that kill or stop the growth of microorganisms including bacteria and fungi. Antibiotics are one class of antimicrobial. It is a larger group which also includes antiviral, antifungal and antiparasitic drugs. So, what are antibiotics? Antibiotics are the chemicals produced by or derived from microorganisms. The first antibiotic was discovered by Alexander Fleming in 1929 in a significant breakthrough for medical science. There are two types of antibiotics. Those antibiotics that kill bacteria are called bactericidal antibiotics. Bacteriostatic antibiotics which can stop the growth of bacteria. There is another classification of antibiotics. First one is narrow spectrum antibiotic. These kill limited range of microorganism only on either gram positive or gram negative bacteria, not both, like penicillin and isoniazid which kills only mycobacteria. Second category is for broad spectrum antibiotics. These antibiotics kill wide range of microorganisms, both gram positive and gram negative. Its example is tetracycline. Let's check the difference between antibiotics and chemotherapeutic agents. Antibiotics are naturally occurring materials while chemotherapeutics are synthesized in the lab. Most antibiotics are now synthesized and are therefore actually chemotherapeutic agents. We use antibiotics in combination because combinations are sometimes used to fight infections. We saw some effects. First one is synergistic effect. It is action of one drug when enhances the activity of another or vice versa, that is known as synergistic effect. In antagonistic effect, the activity of one drug interferes with the action of another. As you can see, this is a picture of antibiotics. These are different classes of antibiotics which is depend on the structure. This one is beta-lactam antibiotics, tetracyclines, aminoglycosides, cyclic peptides, lincosamides, sulfa antibiotics, oxazolidinos, quinolones, macrolide antibiotics. We can also classify antibiotics on basis of mechanism of action. First one is those antibiotics which inhibit protein synthesis. Second category is for those antibiotics which inhibit cell wall synthesis. Third category for those antibiotics which inhibit nucleic acid replication and transcription. Next one is for those antibiotics which inhibit enzymatic activity and synthesis of essential metabolites. Next category is for those antibiotics which injury to plasma membranes. Let us focus on first category of antibiotics, cell wall biosynthesis inhibitors, which are also known as beta-lactams. These antibiotics are bactericidal and they interfere with peptidoglycan synthesis which results in cell lysis. Low toxicity they show and they are active only against growing cells. The examples are penicillin, vancomycin and other antibiotics in the beta-lactam group or in penicillin family, methicillin and ampicillin. Second category for protein synthesis inhibitors. These type of antibiotics were first isolated in 1948 from a Streptomyces species. They inhibit the protein biosynthesis. They are bacteriostatic. They interfere with prokaryotic 70S ribosomes. They also found in mitochondria. They prevent binding of amino acyl transferase RNA to the ribosome. The examples are chloramphenicol, tetracycline, erythromycin and streptomycin. Let us focus on chloramphenicol. Chloramphenicol is widely used because it is inexpensive, easy to manufacture and readily available antibiotics. Originally in 1950s, it was developed naturally from Streptomyces species, but now synthetically manufactured. It is chlorinated nitroaromatic compound. You can see the chemical structure of chloramphenicol here. Chloramphenicol is a bacteriostatic drug that stops bacterial growth by inhibiting protein synthesis. It prevents protein chain elongation by blocking the peptidyl transferase activity of the bacterial ribosome. It prevents peptide bond formation. Let us discuss the spectrum of activity of chloramphenicol. The chloramphenicol is used in the treatment of typhoid because of its excellent blood-brain barrier penetration. Chloramphenicol remains the first choice treatment for staphylococcal brain abscesses. Chloramphenicol is active against the main bacterial causes of meningitis. Chloramphenicol has been effective in treating ocular infections caused by a number of bacteria, including Staphylococcus aureus, Streptococcus pneumoniae and Escherichia coli. But there are some side effects also. The side effects are quite serious. Chloramphenicol may cause bone marrow toxicity during treatment. Its effect manifests first as a fall in hemoglobin levels. The most serious side effect of chloramphenicol treatment is aplastic anemia. Let us focus on tetracycline. Tetracycline are 4 fused C6 rings, 1 benzenoid with variety of substituents and functions. Tetracycline are a family of antibiotics used to treat a broad spectrum of bacterial infections. It includes tetracycline, oxytetracycline, terramycin and chlorotetracycline. This is the chemical structure of tetracycline. Let us discuss the history of tetracycline. Tetracycline was first isolated in 1948 from a Streptomyces species. It is bacteriostatic in nature. Target is protein biosynthesis. Action is it prevents binding of amino acyl transferase RNA to ribosome. The spectrum of activity of tetracycline. Tetracycline are broad spectrum. They kill gram positive bacteria as well as gram negative bacteria and rickettsia, but with weakness Proteus and Pseudomonas. Chlortetracycline, which is naturally origin, doxycycline, it is semi-synthetic by origin. Let us discuss the therapeutic effect of tetracycline. Tetracyclines are used to treat mild acne, upper respiratory tract infections, urinary tract infections, sexually transmitted diseases and typhus. There are some side effects also. The side effects are nephrotoxicity and ototoxicity. It should be avoid use in children because of calcium chelation. Because of that in children yellow teeth have seen. Third category of antibiotics are those antibiotics which inhibit nucleic acid, DNA and RNA. The antibiotics which inhibit the enzyme DNA grase, also called topoisomerase 2. It is an enzyme which is involved in uncoiling supercoiled DNA, normally 1000 micrometer coil to 1 micrometer prior to cell division. These have selective action on prokaryotes, unlike many other drugs acting on DNA. These type of antibiotics interfere with DNA replications, transcription, and it is used for urinary tract infections. The examples of these category of antibiotics are rifampin and quinolones. A major group of broad spectrum antibiotics are quinolones of recent development, which is based on quinoline ring. Let us discuss their origin. They origin from nalidixic acid in 1962. These type of antibiotics are active against gram-negative bacteria and they are used for urinary tract infections. They have some unpleasant side effects also because of poor pharmacokinetic profile and they develop resistance quickly. Their spectrum of activity. The fluorination at position 6 gives much better activity and broader spectrum. Many new analogues being developed with improved activity versus gram-positive bacteria. There is an example of ciprofloxacin. It is effective against many multi-drug resistance bacteria. It is well tolerated, rapidly growing in market and may be best broad spectrum agent now available. Next category of antibiotics, those injury to the plasma membrane. These kind of antibiotics are bactericidal. They disrupt the structure of function of the bacterial membranes which are permeability barrier. The origin of these kind of antibiotics are tyrosidine, gramicidine, polymyxin. These are produced by spore forming gram positive bacteria and this bacteria is Bacillus species. Little systemic use because of toxic side effects. The examples of these kind of antibiotics are many polypeptide antibiotics like miconazole, which is an antifungal agent, polymyxins and polymyxin B, which is an antibacterial drug. Polymyxins originate in 1947 from Bacillus polymyxa. It is the most important clinically cyclic cationic drug and it is fatty acylated. Polymyxins are active against gram-negative bacteria including Pseudomonas aeruginosa and it has some side effects also. Nephrotoxicity limits its systemic use, but still application as topical agent. These are used under closed medical supervision. Let us focus on polymyxin B. Polymyxin B works by attacking the cell membrane of bacteria. It is a narrow spectrum antibiotic. It is not absorbed when taken orally or applied topically. It is used as an ointment or wet dressing. Next category of antibiotics are those antibiotics which inhibit the synthesis of essential metabolites. The antibiotics inhibit the synthesis of folic acid which is necessary for DNA and RNA synthesis comes under this category. These antibiotics have some side effects like crystalluria and skin rashes. The examples of this category of antibiotics are sulfa drugs. Those are sulfanilamides, sulfadiazine which is also known as trimethoprim, sulfadimethozine and sulfadimethozine which is also known as ormetoprim. Let us focus on sulfonamides. Gomek in 1927 began a systematic investigation of gold compound, acridines and azodides. He worked with strain of bacteria from a fatal case of septicemia. He discovered the red diazoprotensil had antibacterial activity and which has low toxicity in mice. He performed a miraculous cure of a 10-month-old baby with staphylococcal infection in 1933 and further he cured his daughter of the same infection in 1935, and he won the Nobel Prize in 1939. Sulfa drugs. Sulfapyridine was shown to be effective against pneumonia in 1938. Sulfacetamide found highly successful use in fighting urinary tract infections which start in 1941. Let's focus on sulfonamides. Sulfonamides are synthetic antimicrobial agents with a wide spectrum encompassing most gram-positive and many gram-negative organisms. These drugs were the first efficient treatment to be employed systematically for the prevention and cure of bacterial infections. Let's discuss about a drug, sulfacetamide. Sulfacetamide is a sulfonamide antibiotic. It is an antibiotic that is used to treat skin infections and urinary tract infections, and it is used to study the treatment of acne, seborrheic dermatitis, neonatal chlamydial conjunctivitis and meningococcal conjunctivitis. This is the chemical structure of sulfacetamide sodium. Next drug is sulfamethoxazole. Sulfamethoxazole is a sulfonamide bacteriostatic antibiotic which is commonly used to treat urinary tract infections and treat sinusitis. Sulfamethoxazole is the drug of choice for pneumocystis pneumonia and which affects primarily patients with HIV. This is the chemical structure of sulfamethoxazole. Let's discuss one more antibiotic, that is trimethoprim. This drug was developed by George H. Hickings and collaborators who shared the Nobel Prize for Physiology or Medicine in 1988 for the discovery of antifolates. It is a bacteriostatic antibiotic mainly used in the prophylaxis and treatment of urinary tract infections. This is the chemical structure of trimethoprim. Trimethoprim was commonly used in 1:5 combination with sulfamethoxazole and it is known as cotrimoxazole. It inhibits an earlier steps in the folate synthesis pathway. Cotrimoxazole is used as prophylaxis in patients at risk for pneumocystis cerevisiae pneumonia. As you can see in this graph, in one side there are number of bacteria in arbitrary units and this side time. We are checking the growth of E. coli. If we do not use any drug, the E. coli will grow faster, you can see in this graph, and if we use trimethoprim alone, the growth is this, and if we use sulfamethoxazole alone, you can see the same graph. But if we use a combination of trimethoprim and sulfamethoxazole at 1:5 together, they show greater inhibition of bacterial growth. This is, you can see, the combination of these two drugs show great inhibition. But there are some side effects of cotrimoxazole also. The combination use has been declining due to bone marrow toxicity, the resistance and lack of greater efficacy in treating common urine and chest infections, and the side effects of antibacterial sulfonamides. Let's discuss the mechanism of action of sulfa drugs. In sulfa drugs, the active agent is sulfanilamide, which is an analog of para-aminobenzoic acid PABA, which is a bacterial growth factor used in biosynthesis of folic acid, which is acquired by man through the diet. Sulfa drugs inhibit normal bacterial utilization of PABA for the synthesis of folic acid, and all cells require folic acid for growth, and it is important metabolite in DNA synthesis. As you can see, this is sulfanilamide and this is PABA. Let us discuss the side effects of sulfur drugs. The common side effects of sulfacetamide and sulfur combination products are local irritation and contact dermatitis. Sulfacetamide should not be used by individuals who have a sensitivity to sulfur or sulfa. Now, let us discuss antihelminthic drugs. Antihelminthic and antiprotozoan and antifungal drugs are more difficult to develop. The reason is these are eukaryotic cells which resembles to human cells. Antihelminthic drugs are used to treat infections with parasitic worms, and this includes both flatworms like flukes and tapeworms and roundworms. There is an example of antihelminthic drug, that is albendazole, which is a broad spectrum antihelminthic drug. Its route of administration is oral. It is neurocysticercosis where surgery is not possible. Albendazole is used. It blocks the glucose uptake by the larva, it depletes glycogen storage of the adult worm and decreases the formation of ATP as a result, and the parasite is immobilized and dies. Let us discuss antiparasitic drugs. Most commonly used antiparasitic drugs are quinine and metronidazole. This drug is used for treatment of various disorders associated with gastrointestinal discomfort, headache, depression, which is not common, and also used to treat peptic ulcer. Let us focus on antifungal agents. Fungi includes yeast, rust, molds and mushrooms cause a range of illness because they have mycosis. It is ranging from the chronic to the serious. These mycoses can manifest themselves in variety of ways. Let us discuss the type of fungal infections. The fungal infections can be superficial and systemic. Superficial infections are at or close to the surface of the skin, and systemic infections affect the body as a whole rather than individual part or organ. These infections are contagious and cause intense itching. Some common fungal diseases are athlete's foot, which is known as tinea pedis, jock itch, which is tinea curis, tinea manis, which is infection of the hand, and onychomycosis, which is affecting the nails. These are examples of superficial infections caused by the dermatophytes from the trichophyton, microsporum, candida. Some can also cause systemic infections here and epidermophyton species. Let us discuss the different types of antifungal agents. First one is imidazole antifungal agent. Second category is for antimetabolic antifungal agents. Third one is polyene antifungal agents, and fourth one is superficial antifungal agents. Let us discuss about imidazole antifungal agents. They work by interfering with the metabolism of RNA and proteins. Its examples are flucytosins, which interferes with DNA and RNA synthesis. It is usually used in combination with other antifungal agents. Second category of polyene antifungal agents. Polyene antifungal antibiotics are produced by the soil bacterium Streptomyces, which work by binding to the fungal cell membrane. It is used with caution because of its side effects, and the polyenes are active against all sterol containing organism, that is yeast, algae, protozoa, flatworms and filamentous fungi. The example of this kind of antifungal agent is amphotericin B. Next category is imidazole antifungal agents. They work by causing leakage to the fungal cell membrane, and its example are ketoconazole. It is a broad spectrum derivative used for systemic fungal infections. Ketoconazole, which is used for superficial infections. Next category is superficial antifungal agents. These are most antifungal agents which act against the cell wall steroid ergosterol, either by affecting its biosynthesis or through interaction with it when it is situated in the cell wall. However, there are two antifungal drugs that affect the fungal cell nucleus. Its examples are grezofulvin and 5-fluorocytosine. Let us discuss the side effects of antifungal agents. These are commonplace but rarely serious, like headache and nausea are most frequent, abdominal pain, diarrhea and vomiting less so. There is evidence that grezofulvin is teratogenic. It causes abnormalities in mice fetuses. Hence, pregnancy is a contradiction for this drug. Now, there is very important point that is known as antibiotic resistance. Let us discuss what is this. When bacteria are exposed to the same antibiotics over and over, the bacteria can change and no longer affected by the drug. Antibiotic resistant bacteria are those germs that are not killed by commonly used antibiotics. Let us discuss how the bacteria become antibiotic resistant. First, bacteria possesses an internal mechanism of changing their structure. So, the antibiotic no longer works, they developed way to inactivate or neutralize the antibiotic. Second way, bacteria can transfer the genes coding for antibiotic resistance between them, making it possible for bacteria never exposed to an antibiotic to acquire resistance from those which have. The problem of antibiotic resistance is worsened when antibiotics are used to treat disorders in which they have no efficacy. Resistance to antibiotics poses a serious and growing problem because some infectious diseases are becoming more difficult to treat. Let us discuss how can we control these antimicrobial resistance. First, it is responsibility of physician. They must work to identify microbe and prescribe suitable antimicrobials. They must educate patients, and then there is responsibility of patients also. They need to carefully follow instructions. They must understand appropriateness and limitations of antibiotics. Antibiotics are not effective against virus. So, viewers, in this session we discussed what are antimicrobial drugs. We classified them on basis of their mechanism of actions, under which category we discussed penicillin, tetracyclines and sulfa drugs, their mechanism of action, how do they work. After that we discussed some antifungal agents and antiparasitic drugs, and after that antibiotic resistance. So, viewers, in next session we will discuss some antiviral drugs, some antiretroviral and anti-leprosy drugs also.